welcome back to my channel. Today we'll be learning about a very interesting topic that is called as AIDS. So before we move on to the session, do not forget to like, share, comment and subscribe. So let us start the session. What is the full form of AIDS? AIDS means acquired immunodeficiency syndrome. Acquired means it develops within the lifetime. AIDS is caused by a very important virus which is called as HIV. HIV means human immunodeficiency virus. So it was first reported in USA in the year 1981. During this period, it was spread almost all over the world, causing 25 million deaths. HIV was first isolated and identified by Professor Montagnier, he is from France, and Professor Gallo from USA. First, it was called as HTLV type 3 virus. It is a former name of the HIV, which is called as human T lymphotropic virus type 3. Later, HTLV type 3 virus was named as HIV human immunodeficiency virus in the year 1986 by International Committee of Viral Nomenclature. Types of virus. Majorly, there are two types of virus. The first one is called as HIV-1 or HIV type 1 virus, which is consisting of 1 to 8 genes. That means the pathogen contains 8 genes, which is almost found all over the world. Then we have HIV type 2 virus, which is consisting of 9 genes. That is, pathogen contains 9 genes, and this type of virus is found only in Africa. Now let us study about the structure of the HIV virus. In terms, we also call it as retrovirus. As you can see on the screen, it is spherical in shape, plus the innermost core is having 2 single-stranded RNA, and this virus is made up of 2 protein coverings. It is called as, the inner covering is called as protein or P24. The outer covering is called as protein or P17. Not to forget about a very important enzyme which is called as reverse transcriptase. This is an enzyme which is responsible for transcription of DNA from RNA. The structure of the virus is covered, or it is made up of an envelope which is mainly made up of lipid. Okay? Plus you can see a spike-like structures or knob-like structures around this structure of this virus. It is made up of glycoprotein and it is of 2 forms, GP120 and GP40.
Modes of HIV transmission. Number 1, when a person is having sexual contact with multiple partners will be at high risk. Number 2, transfusion of contaminated blood and blood products of infected person. Number 3, sharing needles and syringes. Here, intravenous drug abusers will be at high risk. Number 4, during pregnancy or birth. Here, the babies are at the higher risk.
Now, I'll be explaining you about the mode of HIV virus attacking an animal cell. Always remember, virus enters the macrophages or helper T cells. These are the immune cells which build up the immune system of the body. Helper T cells can also be called as T4 cell or CD4 cell. Now, these helper T cells will be having antigen receptor sites. So these antigen receptor sites will help the virus to stick on with the help of glycoproteins. Now let us talk about the mode of action by retrovirus. So already I have told you retrovirus is going to infect first, or it is going to enter the macrophage or T helper cells. Now, once it enters the T helper cells or macrophages, they can directly come and enter into the animal cell. The protein coat will be left outside only the viral RNA will be sent inside, that is inside the plasma membrane of the animal cell. Once the viral RNA is released inside the cell of an animal, now what happens is, since you know that the structure of the retrovirus is having a very important enzyme which is called as reverse transcriptase. What will happen here? The viral RNA is going to change into viral DNA. This is because of reverse transcriptase. You have to remember this. Now, all this viral DNA will enter into the host genome. Whose host here? It is animal. The animal cell genome is now having viral DNA. Now, the viral DNA is going to change into RNA with the help of one important process, transcription. Now, new viral RNA is formed. The multiple copies have been formed. So, it is produced by the infected cell, as you can see here. Now, new viruses are produced. As you can see, the number of viruses are going to multiply. So, these can be released out of the cell and they can infect the other cells also. So, this is the mode of action by the retrovirus. When there is increase in the viral cells inside the body, we are more prone to the infections or certain diseases. This is because there is decreased form of macrophages or helper T cells which were regulating our immune system.
Symptoms of HIV. An individual suffering from HIV can show pneumonia, tuberculosis, fever, diarrhea, weight loss, this is why it is called as slim disease, rashes, ulcer over the whole body, finally leading to brain damage. The time period taken between infection and the appearance of symptoms is 5 to 10 years. Test to detect HIV. ELISA test will be done. ELISA stands for enzyme linked immunosorbent assay. The confirmatory test will be Western blotting. Treatment. There is no cure for AIDS. However, different drugs can be taken to prolong life. Like antiretroviral drugs can be taken, but death will eventually occur. Azidothymidine AZT or zidovudine was the first drug used to slow the progression of disease in patients infected with HIV who have advanced symptoms or no symptoms at all. Few reverse transcriptase inhibitors can be effectively used. The best treatment is prevention.